Okay, so this is your last review lecture for Unit 3. We're going to talk about basically nucleic acids, DNA replication, transcription, translation. All right, so you will remember that DNA and RNA are made up of nucleotides. That's the subunit, right? DNA is double-stranded. RNA is single-stranded. DNA is our genetic information kind of storage found in the nucleus of eukaryotic cells. There's lots of types of RNA. There's tRNA, there's mRNA, there's ribosomal rRNA, and actually a lot of other types of RNA. And these are kind of more temporary nucleic acids. So RNA is transcribed. All of these are transcribed from DNA, and then they have their different functions. So let's remind ourselves about the nucleotides. Let's focus on DNA primarily and base pairing. And one of your objectives is why do pyrimidines, and purines base pair? And I should put like in DNA. And so you're always going to have a base pair of a pyrimidine, which has the smaller one-ring structure, and a purine, which is larger with a two-ring structure. And these all go together because this is how it fits into that double helix space. So if you have two pyrimidines, your helix would actually indent. It's too small. And if you have two purines, it's too wide for the nice double helix structure that Watson and Crick and Rosalind Franklin and others came up with. And remember that A and T base pair with two sets of hydrogen bonds, and G and C base pair with three sets of hydrogen bonds. DNA is anti-parallel, so the two strands run in opposite directions, right? So we have 5' to 3' that base pairs with 5' to 3' going the other way. And why does it have to be anti-parallel? Well, the main reason is so that these hydrogen bonds can line up. So I took some nucleotides and flipped them upside down so that we'd have the 5' to 5', 3', 3'. And you can't get the hydrogen bonds to line up correctly when you make two parallel strands. So DNA is always anti-parallel and then has the base pairing by the hydrogen bonds. So remember, these are weak bonds, but they're strong when there's many of them, so they hold those two strands of DNA together in our double helix structure. All right, so to remind ourselves about the nucleotides, remember that they are made up of a sugar, which is only 5 carbon, versus like glucose is 6 carbon, and that's shown here as this pentose, right? They have a nitrogenous base, which is what we just talked about, the A, G, C, or T, or U if it's RNA, and they have phosphate right over here. Remember that the D in DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid, which means that the deoxy means that DNA has lost an oxygen. So A couple of things to note. Both have 3' OHs, right? That's where the next nucleotide is always joined. DNA has lost an oxygen here. That's how it's different than RNA. And the way we get our 5' to 3' orientation is the numbers 3' are based on how we number the carbons, or the numbering of the carbons on the sugar. Okay. So the 5' carbon is this one right here. It's attached to the phosphate. So the phosphate is always at the 5' end, and the OH group of the sugar is always at the 3' end. And you can see these are on the same side of the molecule, and then the nitrogenous base is on the opposite side. Right, and like I said, DNA, the nitrogenous bases are A, G, C, T. RNA, it's A, G, C, and U. And why is this 3' end so important? So the 3' end is where we attach the next nucleotide. So when you attach the sugar and the phosphate, this is called a phosphodiester bond. Right. What's also kind of cool to see, now that you know more about energy, is nucleotides have 3 phosphates already. So what happens is we break this bond as we connect them. And so nucleotides provide their own energy for DNA replication and for the synthesis of RNA. Another important thing to remember is that DNA polymerase requires a 3' hydroxyl already there to start synthesis. Therefore, we need an RNA primer. Okay. RNA polymerase, which is important in transcription, and primase, important in DNA replication, does not require a 3' OH to start. Okay. So we have a 3' hydroxyl always required to add the next nucleotide, but DNA polymerase needs one right there to start, whereas RNA polymerase can just look at or can just find a strand of DNA and start copying anywhere. All right, so let's look at DNA replication. You're going to notice in your objectives, we're not going through as many details, but the key points that I want you to see from this figure is that replication is bidirectional, which means it goes in 2 directions. So you can see one direction. Change my pen here. Going this way, always 5' to 3'. and the other one going this way, 5' to 3'. And if we looked at a replication bubble where we really talk about bidirectional, we'd be going in two directions, two replication forks, right? This is because DNA polymerase can only add at the 3' end. And because our strands are anti-parallel, we have to go in two directions, which also leads us to the idea of semi-discontinuous replication, which means we have a leading strand up here that's replicated continuously and a lagging strand that's made in kind of chunks, right? It starts, and then as the replication fork opens up, we have to make more, and we have to make more, and we have to make more. The other topic or important point you can see is that DNA replication is semi-conservative, which means that each new double helix is made of one parent and one daughter strand. So one original and one newly synthesized strand. The proteins that I want you to remember about are only a few. First of all, we have, oops, try this way. We have helicase, and we have our single-stranded binding proteins, and these are important for opening up the double helix and keeping it open. So this breaks the hydrogen bonds. And SSB keeps the DNA open. So it's coating the single strand so that it doesn't base pair back together. Another important protein to remember is primase. Let me get a little clearer up here. So primase is an enzyme that makes the RNA primer. And every strand has an RNA primer. So you can see even the leading strand started off with an RNA primer, and every Okazaki fragment here has an RNA primer that it starts off with. Right? Remember that primase can start anywhere. Right? Doesn't have to have a 3' OH to start. So this is providing DNA polymerase the 3' hydroxyl group. All right, the other, okay, so DNA polymerases. We have two, DNA Pol delta, DNA Pol alpha. DNA Pol delta starts at the RNA primer. Just gonna erase some of this. And is the main replication protein or enzyme. Right, so DNA Pol, oops, I didn't change this one. This is DNA Pol delta over here too. That's the main replication. DNA Pol alpha, actually let's just erase some of this. DNA Pol alpha starts actually at this next DNA. So let's say starts at previous DNA fragment and removes the RNA primer. It's trying to show you that right here with little red RNA nucleotides being kicked out. And fills in where the RNA primer was with DNA. So with DNA nucleotides, right? And so DNA poly alpha is going to keep going until it gets right to this spot right here. So it's going to go, go, go. And then, like you can see here, whoops, is that we need ligase to make that final connection of the fragment. So DNA polymerase can add to the 3' end, but then it can't make this last little phosphodiester bond. So ligase comes in and connects the DNA fragments. Okay. I think that's all I want. So let's do a couple questions. Your DNA structure. So again, like always, I encourage you to pause the video at all of these blue slides and test yourself. So this first question asks, what type of nucleotide is this? You should recognize that it's deoxy, so it's got to be DNA. And then this is a purine, because remember it's small name, large structure. So the answer is C. This question asks, where will the next nucleotide be added? The answer is B. This is the 3' OH. Remember, this is the 2'. So this is RNA nucleotide. So we're going to add the phosphate from another nucleotide right here. Phosphate, sugar, base. Next question, name this base pair. So hopefully you see 2 sets of hydrogen bonds, means it has to be an AT base pair. This is A because it's a purine, and remember purines are pure agony, so it'd have to be A or G. And those base pair, A base pairs with T, so that's the pyrimidine. So this is an AT base pair. Which is the 5' end of this molecule? All right, so I've kind of reverse flipped this. And so what you want to always look at is phosphate is where the 5' end. Right? Here's oxygen down here. So the 3' end you see is down here. These are the bases. So the answer is B. Phosphate sugar backbone is always on one side. The bases are always on the other side. Which is the leading strand? The answer is C. Leading strand is continuous and points to the replication fork. Okay, which letter is at a 5' end? This is why I always want you to write 5', draw the arrow to 3'. And so wherever arrowhead is, that's the 3' end. The end of that strand is the 5 prime. Since we have to go anti-parallel, this would be 3 prime, this would be 5 prime. So the answer is B. Okay. If DNA polymerase could copy DNA both 5 prime to 3 prime and 3 prime to 5 prime, which characteristic of DNA replication would no longer be true? The answer to that is the semi-discontinuous. So if DNA, this is our original strand, and DNA can copy both 5 prime to 3 prime and 3 prime to 5 prime, we could just have continuous replication, right? We wouldn't have to remake, like we see up here, all these Okazaki fragments with the leading strand, right? It could just keep on going towards that replication. But that's not how it works. So DNA replication is semi-discontinuous. A mutation in helicase would affect which of the following? Probably should have put this somewhere else so it was a little harder. But we've been talking about DNA replication, and remember that DNA replication uses helicase to open the double strand. Right? Transcription has to open the double strand too, but it uses RNA polymerase, which has its own helicase activity built into it. Okay. RNA splicing translation doesn't have to deal with opening up the DNA strand at all, so there's no helicase activity required for any of that. Which of the following processes requires base pairing of nucleotides? Well, the answer is all of the above. So we know, we've just talked about base pairing for DNA replication. Remember that transcription, you're base pairing DNA and RNA, so that the RNA, we get mRNA that copies the DNA. And in translation, remember that tRNAs base pair with the messenger RNA. That's how the codons are read. So all of these processes require base pairing of nucleotides. Which of the following would allow you to determine whether a piece of nucleic acid was RNA or DNA? So let's just go through these one by one. The presence of a 3' hydroxyl group. Now, that could be RNA or DNA, right? They both have it. The absence of adenine. Well, this could be RNA or DNA because they both use adenine. The presence of thymine. Well, that's DNA only. So that would be one of the correct answers. Whether the sugar of the nucleotides contains a carboxyl group. Oh, well, this isn't even right. There's no carboxyl group in the 2'. If this had said a hydroxyl group, it could be either one. So this isn't going to help you at all. And then if there was the same amount of cytosine as guanine, so we know that C and G base pair together, but that's only in DNA, right? So if you had 2 strands of DNA, you're going to have the same number of cytosines as guanines because they have to base pair. All right, so you're thinking about differences between DNA and RNA, or differences and similarities. Which of the following statements best describes DNA polymerase? So primase, it is an enzyme required to produce a primer. That's done by primase. So that's not DNA polymerase. It is an enzyme that catalyzes the addition of nucleotides to the 5' end. That's wrong. It's the 3' end. It's an enzyme, I'm going down to D, required to glue pieces together. That's not correct. That's ligase. Enzyme required, that requires the presence of a primer, yes, and adds nucleotides to the 3' end. That describes DNA polymerase. All right, I know there's a bunch of questions here. It's not really saying that your exam is going to have more DNA replication questions. It's just I grabbed some that didn't make to the exam and wanted you to think about DNA replication. DNA replication is semi-conservative. This means that DNA replication is D. Produces 2 new DNA double helices, each containing 1 newly synthesized strand and 1 parent, right? So we've talked about if this is your original DNA, then you're going to make new DNA, and that is semi-conservative. 1 old, 1 new. Which of the following statements about the structure of DNA is false? Well, we know that DNA is a double helix. We know that we have base pairing with pyrimidines and purines. We know that A and T base pair and G and C base pair. So the answer is C. The amount of thymine does not equal the amount of guanine, right? So you can have lots of A and T in a DNA and very little CG, or you could have the opposite, but they're all not equal, right? So this is really getting you to think about the base pairing rules. All right, one last one. Which part of the nucleotide to the right, should say below, will be attached to the leading strand? The answer is A. Remember, we're going to have a 3' OH here, and that phosphate is going to attach there to make a phosphodiester bond. All right, let's talk a little bit about transcription. So up here is our central dogma of gene expression or molecular biology, however you want to call it. And transcription is this method of copying some DNA into messenger RNA. And remember that transcription happens the same way, 5' to 3', we draw that arrow. Our enzyme is RNA polymerase, which here has helicase activity to open up that double strand. Everything is going anti-parallel, so our template strand is 3' to 5'. I should do it this way, right? And we're going to make our RNA 5' to 3'. And if you were looking at sequences, the template strand and the RNA are complementary. They're made by, the mRNA is made by base pairing. And why do we do this? Well, it's safer to let this little piece of RNA go out into the cytoplasm and direct synthesis of proteins versus doing anything with our DNA itself. So remember, DNA is like your grandma's cookbook, right? You don't want to mess with those recipes, but you could copy them down and give them to your friends. And if they lose them, that's fine. You still have that original cookbook. So we don't want our genetic information to get messed up at all. RNA will eventually be translated into proteins. And remember, proteins have so many different, what am I trying to say? So many different functions, right? They're enzymes, their structure, their transporters, their signaling. So proteins, I always call them the doers of the cell. Right? So we need this genetic information. We send out a copy to have the functional units kind of made through translation. All right, so for transcription, one of the things you might be asked is to figure out which is the template strand. So again, we've got 3 clickers that you should try. Right. The template strand, the first thing you look for is the RNA strand because it has U's. And then the template strand is going to be complementary. So A and T base pair, U and A, G and C. So the template strand here is C. This would be called the coding strand. Given the following DNA coding strand, what would be the transcribed RNA sequence? So remember, you're going to use base pairing. And anti-parallel. So I always just do 3', 5', and I say A and U and T and A and G and C. So 3' UAC GU AGC is this one. So anti-parallel and base pairing. A mutation in which molecule will have the greatest long-term effect on the cell? That answer would be DNA. So remember, DNA is our genetic material. RNA is made and broken down. Proteins are made and broken down in the cell. So a mutation in the instructions, if you mess up the recipe, you're going to have crappy lasagna forever when you keep copying that, right? So you don't want to mess with your DNA sequence. Okay, then we moved on to translation. And this is a pretty busy slide, but it shows lots of important information. So, whoops, your translation machinery is your ribosome that has 3 sites, E, P, A. Let me do a different color. You can see these are your tRNAs. This is your anti-codon that reads the 3 nucleotide codon of the mRNA. The sites are important in that the E site is where the tRNA is basically going to exit, because in the A site is where the new tRNA comes in, brings that new amino acid, and then in the P site is where we make the peptide bond to join them. And then the whole ribosome shifts over to the next codon. Yep, so the other thing you can see here is how the codon and the anticodon base pair. Codons are always read 5' to 3'. The anticodon is anti-parallel 3' to 5'. Okay. Initiation and termination. What I want you to remember is that initiation starts at the 5' cap. And it scans to the AUG start codon. And once we get that AUG start codon in, then the whole ribosome assembles. We have that first methionine amino acid in the P site, and now we'll start bringing in new tRNAs with their amino acid to the A site, and the ribosome will keep moving, shifting down and reading the 5' to 3' mRNA. When we come to a stop codon, remember a release factor, which is a protein, comes in and everything falls apart. So there's no tRNAs that read the stop codons. When it says stop, you need to stop translating. This free polypeptide is our protein. It's released, it goes and folds and does its job. The ribosome can reassemble on another mRNA and continue doing translation again. Have a copy of the mRNA codon table with you, right? These are 5' to 3' left to right, and it's the mRNA codons. One letter code tells you the amino acids. So have this available. I will not put it on the exam because it's just too big. So have one accessible for you for the final. So let's do a couple problems. Translate the following mRNA. So again, I suggest you pause, work through these, come back. What you're going to do, if it just gives you an mRNA and you're translating, is you're going to scan for that first AUG, and then you're going to read the codons until you come to a stop codon. So AUG encodes for methionine. Then we have isoleucine, AGC serine, histidine, alanine, proline. And then we stop. Don't continue on translating. Translate the following set of tRNA sequences. All right, so remember that tRNA, is read 3' to 5'. That's the anticodon. And it base pairs 5' to 3'. I probably shouldn't draw the arrow that way, it's confusing. For the codon of the mRNA. So the first thing we have to do is we have to do our base pairing, right? So it would be UAC, UCA, UGA, UGG, CAU. But this is backwards. We need to read our codons 5' to 3', so I would rewrite it 5' UAC, GGU. I'm going this way. AGU, ACU, CAU. And then I would go back to my mRNA table, right, and read, and UAC stands for tyrosine, STH, right? So if you don't remember how to do this, UAC, you go back to your chart and you look up UAC, tyrosine. So practice those so you can do those quickly on the exams. A couple more questions. Where, which letter, will the next blank come in? So the answer, this one's a little tricky. I put this opposite. I put 5' here to 3' here. So the correct answer is actually A. The tRNA, so I wrote these sites backwards. This is the APE site. So a tRNA is always going to come in. It's going to come in and the A site, which is towards the 3' end. This, if you look back here on our ribosome, right, 5' to 3' EPA. 5' to 3' EPA. So I flipped it around and put A, still at the 3' end, but on the left side. Just to challenge your brain a little bit. All right, a couple more. Ellen Husk created a new Martian rover explorer. It's collected some unusual material. It shows nucleic acids, but also polypeptides. What part of the cell might this be? So I hope you remember that ribosomes are composed of proteins, which is polypeptides, and ribosomal RNA, which is a nucleic acid. And it's actually the ribosomal RNA that connects those peptide bonds together. Which of the following serves as the translator or intermediary between mRNA codon and an amino acid? And that's your tRNA. So your tRNA, let's look back here, is your translator, right? It's reading an mRNA codon and it's bringing in the amino acid. So it's going from nucleic acid to amino acid language through this tRNA intermediate. Okay. Couple more topics. So we've talked about lipids and we've talked about carbohydrates and we've talked about nucleic acids. So our final macromolecule to remind you about is a protein. Proteins are made of amino acids with this general amino carboxyl R side chain structure. And this is just showing you a few amino acids. And if you look, the top little kind of part of the structure is all the same. This, and this, and this. These are all the R groups. And remember that, and this one's hard to see, but that's here the R group, is that the R groups determine the tertiary protein structure. We're going to look at this in just a minute. Since the top parts of all those molecules are the same, they're not doing much for the structure. What really makes a protein have a different structure is how these R groups all interact with each other as the protein folds. So let's look at protein structure. We have primary structure, which is just the sequence. And then we have secondary structure, which could be alpha helix or beta pleated sheets. And this is made by that NCC backbone and hydrogen bonds. So this is a very common, these are very common structures. What really gives the protein its unique shape, which gives it its unique function, is the tertiary structures, which is the interaction between R groups. And then some proteins will have quaternary structure, where you have two or more proteins coming together to work together. Okay, so here you can see there's actually 4 proteins that come together to form this functional. So I put some proteins are just fine at tertiary structure. They don't interact with other proteins or subunits. Some require quaternary structure. And the final concept with protein structure is just what I was talking about, the interaction of the R groups of the protein, of the amino acids. This is our tertiary or 3rd degree. So we didn't go into this a lot. I'm just using this to point things out. So you can have those R groups, that's what's sticking out here, making ionic bonds or hydrogen bonds or covalent disulfide bridges or just interactions. And so remember that the hydrophobic part or the amino acids that don't like water are going to move to the inside of the protein, and the hydrophilic ones will be on the outside close to water. Kind of like we talked about the rules with lipids, right? Phospholipid, the head is hydrophilic, it likes to be out by the water. The tails that are hydrophobic want to be inside, away from the water. And again, this is all due to whatever R groups you have in your amino acid chain. All right, let's do a couple clickers. So a mutation in the DNA sequence leading to a change in amino acid codon will have the greatest effect on what structure of the protein? And this is kind of a, you know, if this was really a question, I'd make it a short answer where you had to justify. But what I was really looking for in this one is tertiary, right? When you change an amino acid, that means you're going to change the R group interactions. Okay. Yes, you do have a change in the primary structure. You have a change in the sequence. Doesn't usually affect secondary because remember this is due to this MCC backbone, which doesn't change depending on which amino acid you have. And absolutely quaternary structure could be affected if this doesn't fold properly or if there's some kind of amino acid interactions, that structure could be affected as well. I just didn't, this is really a question for helping talk out points, not something you'd see on the test. Tertiary structure of proteins refers to organization and spatial arrangements of where am I? R groups, right? That's where we talk about tertiary. This is really primary. This is quaternary. This is secondary. And this is secondary structure. When 2 polypeptides come together to form a functional protein, this is called, hopefully you remember, quaternary structure, right? When things are interacting. And lamin A, whoops, sorry, is a protein encoded by the LMNA gene. It causes progeria, which is a rapid onset of aging. So these are some children with progeria. So read through this. It talks about all different types of protein structure. And so the answer for this one is all of the above. All right, just a couple more slides to talk about, remind you of the endomembrane system for protein expression. So it'd be great if you paused the video, looked at these different organelles, and described their function and protein expression. The answer is really on the next slide. So it talks about the rough ER for protein synthesis, as well as the ribosomes. The Golgi for modifying and packaging those proteins, Lysosome is part of this. Lysosome is really for breaking down those proteins, so it's not good for protein expression. And we've got transport vesicles and secretory vesicles that help move those proteins around. Protein expression does not always use the endomembrane system. This is something I want you to remember. It all depends on the protein's final destination. So if the protein is going to be part of that endomembrane system that we just talked about, we should put on here the ER. Oh, well, it's already in the ER, but the ER too. Then it's made on a ribosome on the rough ER. If the protein's final destination is something else, like the nucleus, mitochondria, peroxisome, then it's translated on a free ribosome in the cytosol, so it doesn't use the endomembrane system. Last concept, genome versus proteome. I just want you to think about how all of your cells have the same genetic information, but in fact, they are different tissue types. You have muscle, you have heart, you have liver, you have skin, right? Different cell types due to different proteins being expressed. And this is just trying to illustrate that you could have a strand of DNA, and in some cell types, all these proteins are expressed. And in some cell types, only certain proteins. It just depends on what the cell needs. And so that gives you this kind of like nice big picture that you as an embryo started as, oops, an egg and sperm, made this blastocyst, which we didn't talk about, but you don't need to know that term. Made this little embryo, but you have all these different tissues. And you have all these different types of tissues based on which proteins are expressed in the different cell types. Okay, that finishes Unit 3. I hope these lectures help you review for the exam.